First of all, I would like to thank the organizers of this conference for setting up this event and inviting me as a keynote speaker. I will talk about analysis of epitranscriptomics RNA modifications and their analysis by deep sequencing. Nucleic acids like DNA or RNA contains not only boring IECGE or T residues, but also modified nucleotides, which can be formed accidentally, like in the case of DNA, and a few enzymatic modifications of DNA were found, or in RNA, where the majority of RNA modifications are supposed to be formed only enzymatically. Functions of DNA modifications are quite known. These are important for regulation of gene expression or in DNA repair. For RNA modifications, situation is similar. There are many functions which were attributed already to RNA-modified residues, like stabilization of RNA structure, fine-tuning in codon-anticodon interaction, and many, many other functions. While in DNA, chemical damage is quite known, almost nothing is known. Information is very limited about accidental chemical damage of RNA, even if this can be. I will only talk now about enzymatically formed modifications. So what is the reason to modify nucleotides of RNA? Idea is to get additional properties, chemical or physicochemical properties. So this can be created by altering of base pair properties of nucleotides. For example, base pair A, while the methyl group there may be changes in conformation of the uridine and dehydrouridine. In both cases, conformation of this nucleotide will be different from the parental U. And in many cases, reactivity also can be altered. For example, this replacement of the oxygen by the sulfur group will change the activity of the nucleotide, or in the case of 2' methylation, the presence of methyl group at the 2' O of ribose will protect RNA from cleavage under alkaline conditions. And even in some cases, highly hydrophobic residues were found to be attached to RNA, like this aliphatic group, and this will, of course, modify also hydrophobicity. What we know about the presence of RNA modifications in different RNA classes, of course, by studies in 70s and 80s, tRNAs and ribosomal RNAs were massively studied, but even though this information is only available for a limited number of model organisms like E. coli, Saccharomyces cerevisiae, human, Arabidopsis, and some others. For snRNAs and snRNAs, small nuclear and small nucleolar RNAs respectively, those RNAs were also analyzed, but few sites of modification were found in snRNAs and snRNAs, and again, like in tRNA, for tRNA and ribosomal RNA, this information is only accessible for a limited number of organisms. And recent development in epitranscriptomic field concerns, of course, mRNA modification. There are many reports that mRNA contains different modified nucleotides close to the cap here, but also internal modifications like M6A, pseudouridine, M5C, and others. So these modifications are certainly present in different proportions, and profiling of different mRNAs is still in progress. For small nuclear, small RNAs of different types, small interfering RNAs, very little information is available, but it's known that some of them have terminal 2' methylated group, which will protect them from eutailing and degradation. So emerging idea in the epitranscriptomic field is that RNA modification now is a third level of regulation of gene expression at the same importance of in regulation as modification of DNA by M5C and other modified residues, and methylation and acetylation of the histones. It was shown that RNA modification can be reversible for some of them at least, and this opened a way, of course, of regulating of gene expression at the RNA level. So if we take the picture for translation, in translation, all partners of translational machinery, tRNAs, ribosomal RNAs, and mRNAs are modified. They contain modified nucleotides, and this opens a way of regulation in the at the translational level to provide different protein isoforms and thus modify the translatome by itself. It was already shown that translational reprogramming can be made by initiation of translation, but now there are many evidence that RNA modifications in different partners of translational machinery will also affect final proteome. So how to detect and map RNA modifications in different RNAs? So of course, one can use traditional approaches like two-dimensional TLC or HPLC or different other chromatographies coupled or not with mass spectrometry. So this is very powerful methods, but they are not at all throughput, and you need to purify your RNA in a large quantities and a very pure form. Reverse transcription under specific conditions can also be used, like it's illustrated here, for detection of 2' methylated residues. Here you see the pauses formed by reverse transcriptase at low DNTP conditions to detect 2' methylations. But this is still rather heavy and not high throughput. So we are working on deep sequencing-based approaches by coupling of chemical treatment to reveal specific RNA modifications, and this is coupled to appropriate library preparation, sequencing, and of course bioinformatic analysis. So now I will switch to NGS-based detection of RNA modifications here. So detection of RNA modifications is currently a very hot topic in the field of epitranscriptomics. Several approaches were proposed for this analysis. The most popular methods are based on antibody enrichment. So RNA is first fragmented in small pieces of 100 nucleotides in the average, and these pieces will contain some modified nucleotides. So if there are specific antibodies available against RNA modification, these RNA pieces can be selectively enriched by immunoprecipitation, converted to library, and sequenced. After mapping of those fragments to the reference sequence, we will obtain this kind of profile with enrichment peak, broad enrichment peak, which will cover potential modified position. So this method is extensively used for different modified residues, mainly for M6A, but does not provide a single nucleotide resolution. Single nucleotide resolution approaches include either direct chemical signatures made by modified nucleotides in RNA or induced chemical signatures. Direct chemical signatures can be applied when modified nucleotide can block or alter reverse transcription at some point. So this may happen by misincorporation, which will be visible as a mutation, apparent mutation in cDNA, or abortive cDNA sequence. So these abortive cDNAs have to be captured by appropriate library preparation protocol, which allow ligation to cDNA 3' end. And after library preparation and sequencing, this profile can be observed. So there will be a considerable drop of sequencing coverage at the position of modification, and depending on the misincorporation profile, the presence of different nucleotides at this position. So this is a complex reverse transcription signature composed of both abortive cDNA synthesis and misincorporation. Unfortunately, very few modified nucleotides naturally present in RNA can produce these direct chemical signatures. So to reveal others, we need so-called induced chemical signatures. So RT silent RNA modification can be revealed by specific chemical treatment. So this bulky modified residue will stop cDNA synthesis, and this event will be captured by library preparation and sequencing. So in this case, mainly we will see coverage drop and very little misincorporation, if any. So antibody driven enrichment is extensively used in the field, so which will be described here. It has a name MERIP, so for methylated RNA immunoprecipitation. But of course, if antibodies against modified nucleotide are available, it can be applied also to non-methylated modifications. So there are some improvements to the classical MERIP procedure, so it can be converted to so-called MERIP iClip. So this will be a combination between MERIP and cross-linking, covalent cross-linking with RNA by introducing of natural or synthetic residues during RNA synthesis, can be done in vivo and in vitro. And after cross-linking, antibody will be covalently attached somewhere in the proximity of the modified residue. So this provides better resolution and better specificity because more harsh wash conditions can be applied to remove non-specific binding. So these methods are extensively applied for analysis of different modified nucleotides, and in the very first publication about M6A methylome done by MERIP-seq discovered particular clustering of M6A in the beginning of the transcripts. So for RT signatures for detection of RNA modifications, we can use those approaches for detection of first modified nucleotides, which will have altered Watson-Crick age, which will prevent base pairing. This was demonstrated for M1A and for other modified nucleotides like M1G, M3C, M3U, M62A, and some others. So as was shown in the previous slide, in this case, the presence of modified residue will induce misincorporation of misincorporation in cDNA, which will be seen as a mutation or abortive cDNA synthesis. So different RT signatures were described for modified nucleotides. A very simple RT signature was observed in the case of the inosine. Inosine is, of course, modified nucleotide. It is also called RNA-edited nucleotide, but it's typical RNA modification where initially incorporated A is converted to inosine by deamination. So, of course, inosine has different base pairing property comparing to A, and will base pair with C, which will be seen as a mutation. So this is a simple RT signature. Only one nucleotide is incorporated at the place of inosine, and there is no drop in the coverage. For other modified nucleotides like M1A, we will observe complex RT signatures which will be composed both of mutation and abortive cDNA synthesis. So we applied this approach for M1A mapping in different RNAs, and we started with analysis of M1A naturally present in yeast ribosomal RNA. So as illustrated here, there are three possible issues when M1A is present in RNA. Either reverse transcriptase will stop, or it will incorporate a wrong nucleotide, or in some instances, correct base can be incorporated. So these graphs show the coverage in gray and reverse transcription signature in colored bar, incorporation of different nucleotides at the place of two M1A residues present in yeast ribosomal RNA. So the enzymes are known, synthesizing of these residues. So knockout of RRP8 or BMT2 will make disappear one or another M1A in RNA. And of course, in a double deleted strain, both will not be visible. So we also found that RT signature is context dependent. So depending on the surrounding nucleotides around modified site, so specifically N+1 nucleotide will considerably alter composition of misincorporation at the M1 site, M1A site, due to the modulation of the RT properties. So we applied this approach for mapping of 1-methyladenosine in different RNAs, and here we show the analysis of M1A in trypanosomal tRNAs, and we found a certain number of modified residues in those species, and with some limitations. Here you can see that we can even proceed for relative quantification of M1A in different RNAs by analysis of reverse transcription signature. You see here that a proportion of this red and yellow misincorporation increases in when the level of M1A increases in RNA. So what about the presence of M1A in mRNA? Now, there were several reports stating that M1A is present in eukaryotic messenger mRNA, and in those studies, over 1,000 sites were mapped at high resolution, but there were contradictory reports stating that the sites are mostly present in tRNAs, cytosolic tRNAs, like you see here, and very, very few, if any, potential sites can be present in mRNA. So this is still very contradictory. Different consensus sequence were found in some studies compared to the others. Here, this is typical tRNA tipsy loop, which can be modified at position 58 for tRNAs. So how to enhance reverse transcription signature, how to make it more visible? So there are different approaches for that. M6A, which is normally silent in terms of reverse transcription, the presence of M6A can be revealed either by using of reverse transcriptase mutant with a mutation in the active site, and in this case, enzyme will be more sensitive for the presence of M6A and create visible misincorporation signature, or by the use of analog of the substrate, in this case, selenium substituted DTTP, which will block extension at the position of M1A and M6A, and this can be detected also as a drop in the coverage. Another approach was proposed for some, but not for all modified nucleotides in RNA. Some nucleotides in mRNA, M1A, M1G, M22G, and can be specifically demodified, so by removal, by oxidative removal of the methyl group by ALK-B family enzymes or their mutants. So by observing reverse transcription signature in untreated RNA, which is shown here with a visible signature, it should disappear after demodification, and no drop or misincorporation should be visible at this position. This allowed more certain detection of certain modified residues in RNA. So now I will switch to chemically induced RT signatures, and mostly as illustration, I will talk about profiling of pseudouridine by pseudo-U or psi-sec. Reverse transcription signatures for detection of RNA modifications can be used for the nucleotides shown here, like M1A, M1G, and some others like M3C, M3U, where Watson-Crick age of the nucleotide is affected by modification, and those nucleotides will be unable to base pair correctly with a partner. So in this case, this will be shown in a cDNA by misincorporation and visible mutation, or by abortive cDNA synthesis. So simple RT signatures were found for inosine, for example. Inosine is a typical RNA modification, which is also called RNA editing. So this is inosine is formed from A by deamination. So inosine has different properties, base pairing properties compared to adenosine, and will base pair with C, which will be seen as a mutation in the sequencing profile. So this is a simple signature because there is only one nucleotide which is incorporated, and there is no coverage drop. For many others, both events will be found, cDNA with misincorporation visible as mutation, and abortive cDNA synthesis if library preparation allow to capture those abortive cDNAs. So, and in the sequencing profile, misincorporation and coverage drop will be visible. So we applied this approach for mapping of M1A residues by M1A-seq. So as a model, we used yeast ribosomal RNA, which contains two modified M1A residues formed by RRP8 and BMT2. So after reverse transcription, so three possible issues as indicated previously: abortive cDNA, misincorporation, or incorporation of the correct base. So this coverage is shown in gray, and colored bars here and there indicate reverse transcription signature by misincorporation of different nucleotides. So upon deletion of RRP8, you see that this signature disappears. The same for BMT2, and of course they're absent in both knockouts. So RT signature was found to be context dependent, and nucleotide at N+1 position in the sequence will affect mostly the misincorporation. Like you see here, the proportion of colored bars is different depending on the nature of nucleotide at N+1. So we applied this method for quantification of M1A residues, because proportion of misincorporation will depend on the level of modification, and also to the mapping of M1A in different trypanosomal tRNAs, which were not characterized previously. And as you see here, in many tRNAs in the TPC loop, there is a M1A58 which was found. So what about the presence of M1A in the transcriptome? This is still highly contradictory. So there were reports stating that over 1,000 sites of M1A modification was found, in different positions in mRNA with a consensus sequence which is quite GA rich here. And in another publication and from another group, the same analysis revealed only the presence of M1A in tRNAs and very few, if any, sites in mRNAs. And the consensus sequence, like you see here, is totally different. This corresponds mostly to tipsy loop of tRNA, and here it's a GA-rich motif. So there are still many contradictions about M1A presence in mRNA. So can one enhance somehow reverse transcription signature? So this was shown for detection of M6A because M6A is normally RT silent and cannot be detected by reverse transcription signature directly. But if mutations are introduced in the active site of the reverse transcriptase or analog of DNTP, here selenium substituted thymidine is used, in this case, M6A will generate reverse transcription signature either by abortive cDNA synthesis or by misincorporation. So for RT mutant, misincorporation at M6A sites was detected, and in the case of selenium substituted thymidine, there is a coverage drop due to abortive cDNA synthesis. So for some modified nucleotides, like methylated M1A, M1G, and M22G, those nucleotides can be specifically demethylated, so removal of a methyl group by oxidative reaction catalyzed by ALK-B enzyme family. So in the case of non-treated RNA, reverse transcription signature will be detected, and this reverse transcription signature will disappear when RNA is pretreated by demodification enzyme, so there will be normal profile at this position. So comparison of the two allow more certain detection and even quantification sometimes of modified nucleotide. We applied this approach for quantification of M1A residues in RNA, since proportion of misincorporation depend on the level of modification, like shown here by this proportion of red, yellow, and green bars, and also for mapping of M1A in triplenosomal tRNAs, which were not previously analyzed, and we found several tRNA molecules containing M1A at position 58. So the presence of M1A in messenger RNA is still highly contradictory. There were reports stating that over 1,000 sites of M1A can be found in different eukaryotic transcriptomes, and consensus sequence for M1A modification is this GA-rich motif. And at the same time, in parallel, another study found M1A only predominantly in cytoplasmic tRNAs and very few, if any, sites in mRNA. And consensus sequence for modification is completely different. So this should still be clarified by complementary studies. So how can one enhance reverse transcription signature? So this was demonstrated for the detection of M6A, which is normally RT silent nucleotide. So this, to make it detectable by reverse transcription signature, one should modify either reverse transcription enzyme by introducing mutation in the active site, or by the use of analog of DTTP, here selenium substituted thymidine. So in this case, normally RT silent M6A will be detected either as a misincorporation profile with a RT mutant or by the coverage drop corresponding to arrest of reverse transcription due to selenium substituted DTTP. In other cases, there may be a possibility for enzymatic demodification of some modified nucleotide like M1A, M2G, or M22G, which can be oxidatively demodified by ALK-B type enzymes. So when RNA is analyzed directly, those modifications will generate a reverse transcription signature, like you see here, with a drop and a misincorporation. And the signature should, of course, disappear completely when RNA is demodified, so there is no modification anymore at this position. So by comparing of the two profiles, one can deduce the presence of modified nucleotide and ascertain its existence. So chemically induced RT signatures can be used for many different modified nucleotides. There are several methods which were described for that, but I will present only profiling of pseudouridine here by pseudo-U-seq, also called pseudouridine. So detection of pseudouridine is based on the usage of so-called CMCT. CMC stands for derivative of this carbodiimide, and T for toluene sulfonate, which is counter ion. So this CMC group will be reactive with pseudouridine with nitrogen residues here and there. So this method was already extensively used in 60s and 70s, and then, of course, applied in a high throughput version for pseudouridine mapping in RNA. So pseudo-U or pseudo-RNA-seq is based on comparative analysis of treated and untreated RNA samples. So in the presence of CMCT, CMC group will pseudouridine and block primary extension by reverse transcriptase. And this can be compared with untreated sample where this reverse transcription stop should be absent or at least highly diminished. So by comparing the profiles of unmodified and unmodified modified and unmodified RNA, we can deduce the presence of pseudouridine residues at these different positions shown by red lines. This was applied for high throughput mapping of pseudouridylation in yeast and human cells. So in 3 publications appearing the same year, very different number of sites were reported. So from less than 1,000 sites found in one publication to 30,000 in another one, and up to 60 in the third. So there are very modest overlap between common sites. So about 100 sites were found in common. How to explain this discrepancy? This may be related not only for false positives identification, but also false negatives, because everything depends on the calling of modified nucleotides in bioinformatic pipeline used for analysis. So now I will switch to chemically induced signatures and illustrate our work on detection and quantification of 2' methylations. Detection of 2' methylations in RNA can be achieved by at least 3 different and independent methods. They are based either on reverse transcription at low DNTP conditions. It was noticed already in 70s that reverse transcriptase, when concentration of DNTP is very low, about 1 micromolar, can stop at 2' methylated residue in RNA. So this will generate abortive cDNAs, which will be captured by library preparation and sequenced. So the profile will be similar to any RT arresting profiles for modified nucleotides, with a very well visible coverage drop at position of methylation. So the second method, which is based on alkaline hydrolysis, is RiboMed-seq and derived procedures. So this method is based on random fragmentation of RNA under alkaline in alkaline buffer. So this will generate different fragments, and since 2' methylation protects phosphodiester bond in RNA from cleavage, those fragments are showed here, ending with modified nucleotide or starting from nucleotide N+1 will be somehow excluded or at least underrepresented in the library. So RNA will be converted to, these fragments will be converted to the library, sequenced, and counting of 5' and 3' ends for the fragment will allow to establish a profile, and modified nucleotide is visible as this characteristic gap in the coverage. And the third method, which was proposed for detection of 2' methylation, is OxySeq or NM-seq, which is based on periodate oxidation of modified nucleotide at the 3' end of RNA. So to reveal this in long RNA should be first fragmented. It was proposed to make benzonase fragmentation, leaving 3' OH and 5' phosphate. So since benzonase will not cleave at the link between NM and any nucleotide, one should proceed at least for one removal of the nucleotide by oxidation and aniline cleavage. So these cycles can be repeated several times to get enrichment of ligatable NM residues, and other non-methylated ribose will be converted to oxidative products. So after library preparation and sequence, NM 2' methylation can be detected as a signal or at the 3' end. So we are mostly using alkanilin RiboMed-seq in our project, since this method also provides quantitative estimation or measurement of modification level. So RiboMed-seq was successfully applied for very different projects, ranging from analysis of ribosomal RNA, tRNA, small nuclear RNAs, and viral RNAs for HIV, and in all cases was very helpful for quantification and mapping of modified NM residues. Now I will talk about application induced RT signatures for detection of several modified nucleotides like M7G, M3C, dehydrouridine, and even hydroxy 5C by the protocol which is called alkaninin-seq. So detection of M7G was described already a long time ago, and is based on sodium borhydride reduction of M7G, and this reaction will fragilize N-glycosidic bond, which can be easily cleaved to the with the formation of a basic RNA site. A basic RNA sites are known to be cleaved by aniline, with subsequent cleavage at the phosphodiester bond in RNA. So we attempted to use this method for detection of M7G, And indeed, it is possible to induce this cleavage and detect the RT stop corresponding to modified nucleotide. But results, experimentally obtained results for this method were relatively poor because there are very massive cleavages happening everywhere in RNA on the aniline and borehydride reduction. Indeed, one can see a specific signal for M7G, which is shown here for the wild-type strain, and which disappear in the mutant, but this signal is extremely minor. So this method still was successfully applied for analysis of tRNA M7G modification. So, but in the alkaline-seq protocol, we looked at the formation of this 5' end phosphate after the cleavage. So, and the protocol which we developed uses this phosphate for selective ligation of for the modified fragment. So first, alkaline hydrolysis, we found that borohydride reduction is really dispensable, and just simple alkaline hydrolysis will cleave or fragilize M7G sufficiently. So we do a dephosphorylation to remove all possible phosphates at the 5' and 3' ends and induce aniline cleavage at the RNA basic site here. So 5' phosphate shown in red here will be competent for ligation, and only those fragments will be enriched in the library preparation. So after adapter ligation and sequencing, the reads will be aligned at the M+1 nucleotide compared to initially modified M7G and other modified nucleotides detectable by this approach. So as a proof of principle, we applied the method for analysis of yeast ribosomal RNA. 18S ribosomal RNA contains single site of M7G made by BAD23 protein. So as you can see here, we have a single and very strong peak for M7G in the wild-type strain, and this is depending on the aniline treatment, and of course, in the BAD23 knockout, it disappears. High specificity of the approach is also attested by the fact that in other ribosomal RNA, we have only background cleavages observed here in any tested conditions. So sensitivity of M7G detection was tested with mixes between unmodified delta BAD 23 strain and wild type RNA. And as you see here, the signal is still visible until proportion of 2.5% of modified RNA in the total mix. The calibration curve for quantification of M7G is not linear because there is a selective enrichment of 5' phosphate fragments during library preparation, but we are extremely sensitive at even low proportions of M7G in the RNA. So we estimate 2.5 % as a limit for detection, which is quite good for detection of modification. So we established full list of M7G sites in yeast Saccharomyces cerevisiae tRNAs, and we found that some other modified nucleotides like dehydrouridine, which is shown here, also give some measurable signals in alkaline-seq. So we inspected a full list of different tRNAs in yeast, E. coli, and human, and found that M3C, which is present at position 32 and in many tRNAs, at least tRNA serine and tRNA threonine, also gives very nice signal for alkanylin-6. And disappears in the TRM140 strain, which is responsible for methylation of cytidine. So we also inspected different profiles for ribosomal RNA and found that hydroxy 5C, which is only found in bacterial ribosomal RNA, also gives weak but quite detectable signal in alkanylin-6. So, and for dihydrouridine, which was already observed in the previous slides, so knowing that the mechanism of the cleavage involves this opening of the ring of the dihydrouridine ring under alkaline conditions, it's not surprising that in alkanylin-6, this unstable intermediate also decomposes, giving the signal. So we used it for mapping of dihydrouridine modification in different yeast strains because there are several enzymes responsible for dihydrouridine synthesis in tRNA, and found that only these sites only partially modified, those are not modified under our conditions, and those show dihydrouridine presence. So other M7G in other RNAs like mRNAs or other RNAs. There were two reports describing the presence of M7G in human methylome and also in microRNAs. So those methods use enrichment of M7G modified RNA. So the initial step is very, very similar to what we are using in our approach based on Bohr hydrate reduction and then cleavage of the N-glycosidic bond. But then this abasic site is selectively ligated with so-called ARP reagent, biotin-related ARP reagent, to pull out those fragments. So we looked on our data for mRNA and found that indeed we have some signals over background level in human and mouse mRNA, here shown for elongation factor 1, but we believe that these are not enzymatically modified M7G, but probably just a basic RNA sites found formed by spontaneous base loss in RNA, because these signals are quite weak compared to enzymatically formed M7G. So, and another study published recently also did not confirm the presence of M7G in mRNA. They used Bohr hydrate reduction and reverse transcription signature formed here for modification because without cleavage, the absence of modified nucleotide will, of course, provoke misincorporation. So what is the take-home message for this part? Of course, we need validation of all experimentally obtained data set and how to proceed for this validation. So the very good idea to use biological and technical replicates or complement the study by using of the mutants or knockout or organisms, or eventually, if possible, to use orthogonal validation approaches like LCMS or the method which is called Scarlet, which is based on selective labeling of RNA. So it would be important also to optimize all bioinformatics steps of treatment. So by cleaning also and optimization of the reference sequences to avoid misalignment of experimentally obtained reads, and also calculation of a random background and statistics for detection for calling of modified sites. In many occasions, validation can be also complemented by careful manual inspections of the profiles to avoid wrong interpretations. And we need careful validation by comparison of different data sets, because only in few cases this comparison was properly done. And so in our lab, we are doing analysis of RNA modifications, of course. So this is frequently complemented by analysis of transcriptomics, because modification may affect so gene expression, and ribosome profiling to understand functions of RNA modifications in different systems. So it remains to thank people who participated in these different projects, mostly in RNA RNP team in our lab, in very tight collaboration with RNA-seq core facility, which is headed by Virginie Marchand, and a collaboration with our partners in Germany, Mark Helm and Mainz University, and in Belgium, Denis Lafontaine. Thank you for your attention.